Fibrosis is a disease characterized by excessive collagen accumulation, causing organs to harden and lose function progressively. PBP1710, developed by IDC, presents new therapeutic possibilities by targeting the root mechanisms of collagen overproduction. Let's hear from the researchers who developed this groundbreaking patent.
When our bodies sustain an injury, they naturally produce proteins like collagen to repair damaged tissue. However, when this healing process becomes prolonged or excessive, normal tissue hardens, a condition known as fibrosis. As fibrosis progresses, organ function deteriorates, and in severe cases, it can become life-threatening. Recent research has identified a protein called CTHRC1 as a critical factor in the fibrosis process. CTHRC1 is highly expressed in fibrotic cells and acts as a functional mediator that actively promotes fibrosis, rather than merely serving as a biomarker. Studies have shown that higher CTHRC1 levels correlate with more severe lesions and reduced survival rates. Therapeutic efficacy of Prestige Biopharma's anti-CTHRC1 antibody, PBP1710, has been demonstrated in animal models of fibrosis. In models of fibrotic diseases affecting kidneys, liver, and systemic sclerosis, CTHRC1 expression was significantly elevated compared to normal mice. Administration of PBP1710 to inhibit CTHRC1 activity demonstrated effective alleviation of tissue fibrosis. Currently available fibrosis treatments demonstrate limited efficacy and are associated with significant side effects. In contrast, PBP1710 is an antibody therapy that precisely targets CTHRC1, the core protein driving fibrosis, potentially offering a more favorable safety profile. We have confirmed PBP1710's effectiveness across multiple fibrosis disease models, and we anticipate that PBP1710 could become a valuable new treatment option for fibrosis when applied clinically.
This patent presents new treatment options to slow fibrotic disease progression and improve patient prognosis. From a therapeutic perspective, PBP1710 monotherapy shows promise in slowing the rate of fibrosis progression. Combined with approved fibrosis treatments, PBP1710 offers potential for synergistic therapeutic benefit. Additionally, this represents significant technological advancement. The antibody-based therapeutic modality concurrently resolves critical limitations of conventional small molecule compounds, namely lack of target specificity, adverse side effects, and difficulties with chronic administration. Furthermore, this technology is not confined to a single disease, but applicable across fibrotic conditions affecting multiple organs, including the kidneys, liver, and lungs, offering broad clinical utility. Ultimately, this patent represents an opportunity to reframe fibrosis treatment as a comprehensive strategy, targeting common underlying mechanisms rather than treating individual diseases in isolation. It represents a meaningful starting point for establishing a new direction and paradigm in the field of fibrosis therapeutic development.